Good day, everyone. So, for today, we are going to talk about your nucleotide. So, before we go into a deeper discussion about this topic, I'm going to give you the basics of your nucleotide. So, this will going to guide you on further understanding your nucleotides. So, when we talk about your nucleotides, basically, this is composed of your nitrogenous base plus a pentose sugar combined with your phosphate. As compared into your nucleoside, so nucleoside is composed of nitrogenous base plus your pentose sugar only. Okay? Please remember nucleoside, no? Side, it is composed of a base plus a sugar. Okay? Nucleotide, tatlo, no? Tide, nitrogenous base, a sugar, and a phosphate. Okay? So, when we talk about your nitrogenous base refers to the nitrogen containing heterocyclic ring structure. Okay? So, there are two types of your nitrogenous bases. Please remember again, two types yung nitrogenous bases nyo. We have your purine and your pyrimidine. So, under your purine, we have two. This is your adenine and your guanine. For pyrimidine, we have three. We have your cytosine, your uracil, and as well as your thymine. Okay? So for you not to forget the examples of your purine and pyrimidine, so the mnemonics that I'm using for the longest time is your puga for purine, G for guanine, and A for adenine. Okay? And also for pyrimidine, no, what I use is cut the pie, C-U-T, the pie or pie, no? So C for cytosine, U for uracil, T for thymine, pi for your pyrimidine. So puga and cut the pie. Okay, what I want you to remember is that your adenine and guanine, no, are both present in your DNA and as well as in your RNA. No? Again, your adenine and guanine are both present in your DNA and RNA. Okay? Cytosine also is present in your DNA and RNA. However, your uracil is present only in your RNA and thymine is present only in your DNA. Okay, please remember that. So now let's go to your pentose sugar. So let me first erase this one. When we talk about your pentose sugar, there are two types of your pentose sugar. Again, for pentose sugar, there are two types. No, we have your RNA and your DNA. So please remember that your RNA is composed of your D-ribose sugar or your ribose, no? For DNA naman, it is composed of your 2-deoxy D-ribose or your deoxyribose. Okay? So now let us further talk about your nucleoside. Okay? For nucleoside, 'di ba? If you can remember, sinabi natin kanina that your nucleoside refers to the combination of your nitrogenous base and your pentose sugar only. So, sino yung nagdidikit or the bond between the base and the sugar? So, please remember that the one that is responsible for the bonding between the base and the sugar for your nucleoside is your N-glycosidic linkage. Okay? So, pinagdidikit nito si nitrogenous base nyo and si pentose sugar. Okay, now for your nucleotide, we talk about this one earlier that this refers to the combination of your nucleoside plus your phosphate. Bakit nucleoside plus phosphate? Because remember that nucleoside is composed of nitrogenous base plus pentose sugar. And the composition of your nucleotide is nitrogenous base plus pentose sugar plus phosphate. That's why your nucleotide refers to the combination of your nucleoside plus your phosphate. So, di ba I told you earlier that your N-glycosidic linkage will going to serve as the bond between the base and the sugar in your nucleoside. So, now naman, sino kaya yung nagsaserve as a bond between your pentose sugar and your phosphate? So, it will be your ester bond. So, let's try to draw it. So, in your nucleotide, so, di ba sabi natin it is composed of your nitrogenous base. So, ang nagko-combine between your nitrogenous base and your pentose sugar is your N-glycosidic bond, 'di ba? So, it is between your nitrogenous base and your pentose sugar. And this structure is your nucleoside. So, if you combine that, this pentose sugar with your phosphate, sabi natin, the bond is your ester bond. And this whole structure is your nucleotide. So, what will be the purpose of these no, nucleotides? Please remember that nucleotides are the building blocks of your nucleic acids. Again, nucleotides are the building blocks of your nucleic acid. So, other functions of your nucleotides, pwede siyang maging carriers of activated intermediates in the synthesis of your carbohydrates, lipids, and also your proteins. Pwede rin siyang maging structural component of your coenzymes, and also pwede siyang maging synthetic analogs for your drugs. And ano pa, pwede rin siya maging second messengers in signal transduction pathways. But what I want you to remember is your ATP. 'Di ba? ATP is our energy. So ATP is the most abundant free nucleotide in mammalian cell. Again, ATP is the most abundant free nucleotide in your mammalian cell. So sabi natin 'di ba that your nucleotides are the building blocks of your nucleic acids. So what are your nucleic acids? So nucleic acid are the polymer of your nucleotides, and it is joined by your 3' to 5' phosphodiester bond. Again, your nucleic acids are polymers of your nucleotides joined by a 3' to 5' phosphodiester bond. And we have two types of your nucleic acids. We have your deoxyribonucleic acid or your DNA. And also we have your ribonucleic acid or your RNA. So now let's just compare your DNA and your RNA. Okay, so for DNA, so this is your DNA compared with your RNA. So DNA again can be seen in your nucleus. Your RNA is located in your cytoplasm. Your bases, again, for DNA, we mentioned it a while ago. We have your thymine and also your cytosine. For RNA, we have your uracil and also your cytosine. Again, DNA, thymine. For RNA is your uracil. Okay, sugar is deoxyribose. for your DNA and ribose for your RNA. So usually your DNA is double stranded. However, your RNA is single stranded. Okay? So ano pa yung importanteng malaman nyo before we go into our discussion? So importante din malaman nyo as you go on into this topic is your base pairing rule. So for the base pairing rule, so adenine, so your adenine is always paired with your thymine. This is in the case of your DNA. However, if the given is RNA, no, syempre your adenine will be paired with your what? Uracil. Okay? Again, please remember, adenine always pairs with your thymine. However, in RNA, it will be paired with your uracil. For guanine naman, so guanine is always paired with your cytosine. Again, no? So, adenine for thymine. In the case of your RNA, it is paired with your uracil, and guanine is always paired with your cytosine. So, GC and your AT. So, pwede rin naman na AU. Okay? So, there are three scientists who work together on studying the structure of your DNA. So, sino-sino sila? Si James Watson, Francis Crick, and Rosalind Franklin. The proposed model was based on the X-ray diffraction photographs of DNA taken by Rosalind Franklin. Watson and Crick won a Nobel Prize in 1962 for their discovery. So, short trivia, despite the efforts of Franklin, hindi siya nanalo ng Nobel Prize because she had died of ovarian cancer in 1958. And the Nobel Committee does not consider posthumous candidacies. So, ano ba yung features of the Watson-Crick DNA model? So, for the composition, it is a right-handed double-stranded DNA helix. Meaning to say, right-handed means that the DNA strands turn clockwise when viewed along the axis. So, we have also base pairing rule, no? So, ano yung base pairing rule? So, your adenine always pair with your thymine. Okay? And in guanine naman, guanine should always pair with your cytosine. Please remember when it comes to the hydrogen bonding, GC bonding is composed of 3 hydrogen bonds, while your AT bonding is composed of 2 hydrogen bonds. So, meaning to say, no, GC bonding is stronger than AT bonding, which means that the higher the GC content of a DNA, the higher the melting temperature of DNA. So, tandaan nyo lang, pag yung couple ay GC, so graded conscious or grade conscious, they are stronger because they help each other out to have high grade. So, GC bonding, 3 hydrogen bonds. AT bonding, 2 hydrogen bonds. Pag mataas yung GC content ng DNA, higher ang melting temperature ng DNA. Okay? So, with regards to orientation of the two strands are anti-parallel. So, one strand runs in a 5' to 3' direction, and the other strand runs in 3' to 5' direction. So, with regards to the grooves of the DNA, we have two. We have your major and minor grooves. So, these grooves often acts as a site of DNA protein interaction needed for the regulation of gene expression. Okay? So, DNA protein interaction is via your hydrophobic interaction and ionic bonding. Okay, now let's talk about your DNA levels of organization. So, with regards to levels of DNA organization, so again, DNA is double helix. So, it is composed of 10 nanometer chromatin fibril, also 30 nanometer chromatin fibril, and supercoiled structure, and meron din siyang chromosome. Okay. So first, let's talk about the DNA double helix, the 2 nanometer DNA double helix. Okay? So 2 nanometer DNA double helix is the first level of organization of the DNA. Next is your 10 nanometer chromatin fibril. So your 10 nanometer chromatin fibril is consists of nucleosomes, which is separated by linker DNA. So your DNA is being wrapped 1.75 times over a histone octamer in a left-handed helix. So this one is responsible for beads on a string appearance of your DNA on your electron microscopy. Next is your 30 nanometer chromatin fibril. So your 30 nanometer chromatin fibril is a group of nucleosome. And your supercoiled structure promotes the packing of DNA into a compact structure. Also, we have your chromosome. So, the condensation of DNA occurs during prophase of the mitosis. We have 23 pairs of chromosome, 46 chromosomes in total. So, 22 pairs of autosomes and 1 pair of sex chromosomes, your X and Y. Now let's talk about the structural forms of DNA. So there are different types of DNA. 6 types have been identified. Your A, B, C, D, E, and Z. But the three more prominent types are your aDNA, bDNA, and zDNA. Please remember that all types of DNA are right-handed except for your zDNA, which is left-handed. Okay, let us compare your A, B, and zDNA with regards to number of base pairs per turn. Okay, base pairs per turn. So for aDNA, 11 base pairs per turns. For bDNA, 10. And for zDNA, 12 base pairs per turn. With regards to morphology, as you can see here in this figure, your aDNA is broad and short. Your bDNA is longer and thinner. And your zDNA is also longer and thinner. Based on the screw sense, nabanggit na natin, no, that your A and B DNA, both of them are right-handed while your ZDNA is left-handed. So, ano yung features niya? So, your aDNA is found in low humidity and high salt condition. While your bDNA is the most common form physiologically. Again, bDNA is the most common form physiologically. And lastly, your zDNA is seen in 5' end of your chromosomes. Okay? Another important thing that I want you to remember with regards to your zDNA, it is found in areas of alternating purines and pyrimidines. Now let's talk about your nucleotide. So your nucleotide is composed of your phosphate group, your cyclic 5 carbon pentose sugar, and a nitrogen containing base. So wag malilito, again, nucleotide is composed of a nitrogenous base plus a pentose sugar plus phosphate. As compared to your nucleoside is composed of nitrogenous base plus pentose sugar lang. Okay? So, ano ba yung mga nitrogenous base? So, nitrogenous base refers to nitrogen-containing heterocyclic ring structure. So, there are two types of nitrogenous bases. We have your purine and pyrimidines. So, we have here the structures of your purines and pyrimidines. So, for purines, we have adenine and guanine. For pyrimidines, we have cytosine, thymine, and uracil. So, para di ako malito, ang palatandaan ko dito is pure as gold. So, pure as gold. Purine for adenine and guanine. And also, isa ko pang palatandaan is cut the pyramid. So, bakit cut the pyramid? CUT, cytosine, uracil, and thymine, pyramid, pyrimidines. Okay? Pure as gold and cut the pyramid. Please remember, no, kung saan nyo lang sila makikita, your uracil is found only in your RNA, for thymine, DNA only. Again, just to reiterate, note that there are 3 hydrogen bonds between your GC bonding, so your cytosine and your guanine, and just 2 hydrogen bonds between your adenine and thymine. Thus, your DNA strands with a greater proportion of GC nucleotide pairs will be more difficult to separate. Now, let's talk about DNA replication. So, DNA replication occurs in 5' to 3' direction in continuous and discontinuous fashion. It is semi-conservative. Please remember that DNA replication is more complex in eukaryotes than in prokaryotes. Sabi natin that DNA replication is a semi-conservative process. So meaning to say, each strand serves as a template for complementary daughter strand. And each strand also becomes part of the daughter strand. Semi-conservative meaning yung isang strand ay original from the parent molecule talaga, yung isa kagagawa lang during replication process. In mitosis, the cell divides, and please remember, in S phase, DNA is being synthesized. Before we discuss the DNA replication process, let's take a look sa overview muna. So, we have your origin of replication, this one. So, origin replication, particular consensus sequence in genome where DNA replication begins or kung saan nag-start yung DNA replication may be single prokaryotes or multiple eukaryotes. You also have your replication fork. Your replication fork is a Y-shaped region along DNA template where leading and lagging strands are being synthesized. We also have your helicase. So helicase unwinds DNA template at your replication fork. So helicase halves DNA. So we also have your single-stranded DNA binding protein. So, single-stranded binding protein, ang function nito is to prevent strand from reannealing or degradation by your nucleases. We also have your DNA topoisomerase. DNA topoisomerase create a single or double-stranded break in the helix to add or remove your supercoils. Okay, we also have your primase. So your primase makes an RNA primer on which your DNA polymerase 3 can initiate your replication. So ano yung DNA polymerase 3? DNA polymerase 3 is found only in your prokaryotes. So, ang ginagawa nito, it elongates leading strand by adding deoxynucleotides to the 3' end and elongates your lagging strand until it reaches your primer of preceding fragment. We also have your DNA polymerase 1. So, DNA polymerase 1 is found only in your prokaryotes. And ang ginagawa nito, it degrades RNA primer and replaces it with DNA. We also have your DNA ligase. Your DNA ligase catalyzes the formation of your phosphodiester bond within a strand of double-stranded DNA. Now, let us discuss the different steps in your DNA replication. So, first, the origin of replication or your ORI is recognized by a group of proteins called origin recognition complex. In this figure, you can see an enzyme called helicase. So helicase will going to break the hydrogen bonds between the bases of two antiparallel strands, and the strands are initially split apart in areas that are rich in 80 base pairs, forming a replication fork. So we also have your single-stranded DNA binding proteins which will going to maintain the separation of your parent strand. And also we have your topoisomerases. So these topoisomerases will going to relieve torsional strain that results from helicase induced unwinding. So next, your RNA polymerase or your RNA primase synthesizes short RNA nucleotide sequence that acts as primers or starters. These essentially provide a starting point for DNA replication. Now, as you can see in the figure, your DNA polymerase 3 can now start synthesizing the new DNA strand using free DNA nucleotides. However, DNA polymerase can only read the original template in a 3' to 5' direction, making DNA 5' to 3'. So this is not a problem on the leading strand because the DNA polymerase can simply continue to read along as the two parent strands continue to unzip. Again, what is the function of your DNA polymerase 3? So your DNA polymerase 3 elongates the DNA strand by adding new deoxyribonucleotides. Please remember that your DNA polymerase cannot start DNA replication on its own. Thus, it requires your RNA primase. And it can only synthesize DNA in one direction, that is 5' to 3' direction. So on the lagging strand, DNA polymerase moves away from the replication port. And as the strands continue to unzip, more DNA is being exposed and new RNA primers must be added. As a result, the lagging strand is synthesized in short bursts as DNA polymerase synthesizes DNA in between each of the RNA primers. So, the newly synthesized lagging strand now consists of both DNA and RNA fragments, and the DNA fragments are known as your Okazaki fragments. So, next, your DNA polymerase 1 will going to remove the RNA primers. So, aalisan niya tong mga RNA primers na to and replaces them with DNA. So again, when another primer is reached, the DNA polymerase 1 will going to remove the ribonucleotides using your 5' to 3' exonuclease. So lastly, your DNA ligase will go into seals the nick by catalyzing the formation of the last phosphodiester bond requiring hydrolysis and ATP. So let us summarize the last steps of your DNA replication. So again, your RNA primer is being excised by DNA polymerase 1. And then the gap is being filled by DNA polymerase 1. Then afterwards, yung mga remaining nick is being sealed by DNA ligase. Now let us discuss DNA repair mechanism. So we have first your non-homologous end joining. So in here, it brings together two ends of your DNA fragments to repair double-stranded breaks. Well, as you can see here in the figure. So homology is not required and part of the DNA may be lost or translocated. Again, in non-homologous end joining, this one results in loss of DNA. Now let us go to homologous end joining. So in homologous end joining, it requires two homologous DNA duplexes. A strand from a damaged double-stranded DNA is being repaired using a complementary strand from intact homologous double-stranded DNA as a template. So saan natin to nakikita? Nakikita natin to in cases of breast cancers and ovarian cancer as well. Also, in some cases of anemia, specifically in Fanconi anemia. Another thing that I want you to remember is that your homologous recombination or your homologous end joining is less prone to error. So, ano yung mga different causes ng double strand DNA repair? So, we have, di ba, your non-homologous and homologous end joining. So, we have first your ionizing radiation. We also have free radicals and also your anti-tumor drugs. Now let's go to single-strand DNA repair. So first we have your nucleotide excision repair. So in nucleotide excision repair, specific endonucleases remove the oligonucleotides containing your damaged bases. So DNA polymerase and ligase will going to fill and reseal the gap, this gap, no, respectively, and will going to repairs bulk helic distorting lesion. For example, your pyrimidine dimers. So, please remember that your nucleotide excision repair occurs in G1 phase of your cell cycle. So, example kung saan natin nakikita tong nucleotide excision repair, nakikita natin to in your xeroderma pigmentosum or in your XP. So, in xeroderma pigmentosum, nagkakaroon ng inability to repair your DNA pyrimidine dimers caused by your ultraviolet exposure or your UV exposure. So, Xeroderma pigmentosum presents with dry skin, photosensitivity, and as well as skin cancer. Again, the problem in your XP or Xeroderma pigmentosum is inability to repair DNA pyrimidine dimers caused by ultraviolet rays exposure. Now let's go to your base excision repair. So in your base excision repair, the base specific glycosylase will going to remove your altered base and will going to create an AP site or your apurinic or apyrimidinic site, and one or more nucleotides are removed by AP endonucleases which going to cleaves at 5 end, and AP lyase will going to cleaves at your 3 prime end. So DNA polymerase B will going to fill the gap and your DNA ligase will going to seal it. Again, in summary, no, your nucleotides are being removed by your AP endonuclease which going to cleave at your 5' end, and your apelase will going to cleave at your 3' end. And this one, no, ang responsible for filling and sealing the gap na nangyari is your polymerase and your DNA ligase will going to seal it. Okay, so your base excision repair occurs throughout cell cycle, and this one is important in repair of spontaneous and toxic deamination. Now let's go to your mismatch repair. So in mismatch repair, your mismatch nucleotides and newly synthesized strands are being removed and gap is filled and eventually being resealed. So this one occurs predominantly in the S phase of cell cycle. So this one, nakikita natin to in cases of your Lynch syndrome or your hereditary non-polyposis colorectal cancer. Again, Lynch syndrome or your hereditary non-polyposis colorectal cancer or HNPCC. So again, in summary of your single-strand DNA repair, so in your nucleotide excision repair, no, ang trigger nito is exposure to UV light. So your UV-specific endonucleases recognizes the dimer and removes the damaged strand, and eventually your DNA polymerase synthesizes new DNA and your ligase will going to seal the nick. So we can see this one in cases of your xeroderma pigmentosum. Next is your base alteration or base excision repair. So ano yung trigger ng base excision repair? The trigger for this one is spontaneous or presence of deaminating compound. So in here, no, abnormal bases are being recognized and removed by specific glycolases, leaving an AP site. Now let's go to your mismatch repair. So specific cause of your mismatch trend is errors that escape your proofreading. Again, we can see this one in your Lynch syndrome or your hereditary non-polyposis colon cancer. Now, let's go to mutations in DNA. So, mutations in DNA, pinag-uusapan natin dito is the degree of change. So, we have your silent, your missense, nonsense, and frame shift mutation. So, in here, no, single nucleotide substitutions are being repaired by your DNA polymerase and also by DNA ligase. So, types of single nucleotide or point mutations are your transition. So, in transition, this is your mutations in purine to purine or pyrimidine to pyrimidine. Pag sinabi namang transversion, so this one naman is purine to pyrimidine. Example is your A to T or pwede rin naman pyrimidine to purine. So like For example, C to G. Again, in transition, no, same lang, no, purine to purine, like A to G, or pyrimidine to pyrimidine, like C to T. Okay? So, first we have your single nucleotide substitution. So, in single nucleotide substitution, we have your silent mutation, your missense mutation, and your nonsense mutation. So, in silent mutation, it codes for same or synonymous amino acid. Like, as you can see here, no, A to A. Okay? So, it often involves third position of the codon. So, this third position of the codon is what we call your tRNA wobble. Next is your missense mutation. So, in missense mutation, this one naman results in change in amino acid called conservative if new amino acid has similar chemical structure. Example of which is your sickle cell disease. So, sickle cell. So, in sickle cell disease, nagkakaroon ng substitution of your glutamic acid with valine. Again, substitution of your glutamic acid with valine. Next is your nonsense mutation. In nonsense, no, results in early stop codon. So, ano ba yung mga stop codon natin? Example of your stop codon is your uga, uwa, and uwag. So, uga, uwa, uwag. These are your stop codons. So, usually, in nonsense mutation, they generate non-functional protein. So, now let's go to other type of mutation. We have your frameshift mutation and splice mutation. So, in your frameshift mutation, ang nangyayari dito, nagkakaroon ng deletion or insertion of any number of nucleotides not divisible by 3. So, nagkakaroon ng misreading of all nucleotides downstream. So, protein here may be shorter or longer and its function may be disrupted or altered. So, saan natin nakikita itong mga frameshift mutation? Frameshift mutation can be seen in diseases like your Duchenne muscular dystrophy. Also in cases of your Tay-Sachs disease. Now let's go to your RNA or ribonucleic acid. So your RNA is a polymer consisting of purine and as well as pyrimidine ribonucleotides linked together by 3' to 5' phosphodiester band. So compare lang natin siya with your DNA. So sugar moiety, so in DNA we have your deoxyribose. In your RNA, we have your ribose. Purine component of your DNA is adenine and guanine. So, same lang siya with RNA ng purine components. Adenine and guanine lang din in your RNA. With regards to your pyrimidine component naman, in your DNA, we have your cytosine and thymine. In your RNA naman, we also have your cytosine, pero wala siyang thymine, but meron siyang uracil. So, structure of DNA, your DNA is a double-stranded, while your RNA is a single-stranded. So, talking about Chargaff's rule, Chargaff's rule applies only in your DNA. Pero di siya nag-a-apply with your RNA. Talking about stability, mas stable ang DNA and your DNA cannot be hydrolyzed by alkali due to the absence of your 2' hydroxyl group. But your RNA is unstable and can be hydrolyzed by alkali to 2' to 3' cyclic diester of your mononucleotides. Remember, no, RNA, the sugar is ribose, which is a less stable. RNA is also single-stranded, so your Chargaff's rule don't apply, and all RNA virus are single-stranded except for Rio virus. Now let us discuss the different types of your RNA. We have your protein coding RNA and non-protein coding RNA. So first, let's talk about your mRNA or your messenger RNA. So the function of your mRNA is that it copies genetic information from DNA and serves as the template for your protein synthesis. So your messenger RNA is the most heterogeneous RNA. Again, the function of your mRNA is it serves as a messenger conveying information from your DNA to the translation machinery, which is your ribosome. Next is your ribosomal RNA. Your ribosomal RNA is the most abundant RNA. So 80% of the total RNA. And this one is responsible for translation. Again, pag sinabi natin translation, translation is the formation of your proteins. So, in your prokaryotes, we have 50S and 30S subunits, while in your eukaryotes, we have 60S and 40S subunits. Now let's talk about your transfer RNA. So your transfer RNA serves as an adapter molecules that translate the nucleotide sequence of your mRNA going into a specific amino acid. So it contains significant proportion of your nucleotides with unusual bases like your dihydrouridine, your pseudouridine, and as well as inosine and ribotymidine. Next is your small nuclear RNA. So your small nuclear RNA has a functions for gene expression, regulation. So example of which is your mRNA processing and rRNA processing. Next is your microRNA or small interfering RNA. So your microRNA is responsible for gene expression regulation by targeting your mRNA with several distinct mechanisms. And lastly, we have your long non-coding RNA. So this one is responsible for modulation of gene expression in many different mechanisms. Now let us briefly discuss the different steps in your RNA synthesis. We have initiation step, elongation step, and termination step. In initiation step, your RNA polymerase will going to bind with your DNA promoter sequence. So in prokaryotes, we have 35 sequence, your TT, GACA, and your Pibno box about 10 BP. And also in eukaryotes, we have your tata or your Hagnes box, which is about 25 BP. And also we have your CE80 box, which is about 70 to 80 BP. So, in your eukaryotes, the unwinding of your DNA occurs to create a transcription bubble. So, next one after your initiation is your elongation. So, in elongation, ang nangyayari dito is nagkakaroon ng local unwinding of DNA continuous in the five prime to 3 prime direction. So your RNA polymerase synthesizes your RNA using your nucleoside monophosphates, releasing phosphate each time. And last step is your uh, termination. In here, nagkakaroon ng elongation of RNA which continues until a termination signal is being reached. So in termination, we have your raw independent and raw dependent. In your raw independent, this one requires formation of your hair loop. While in your raw dependent naman, it requires raw protein to release RNA from your DNA. Now let's talk about your lactose operon. So your lactose operon is a part of your bacterial chromosome that controls the synthesis of enzymes involved in lactose metabolism. So we have your Z gene, which encodes your beta galactosidase, your Y gene that encodes for galactoside permease, which is a transport protein required for the entry of your lactose in the cell. Also, we have your A gene that encodes thiogalactoside trans-acetylase whose function is still unknown. We also have your I gene that encodes a lac repressor protein that is constitutively expressed and located at a distant site in your DNA. So, full expression of the lactose operon requires your repressor protein must not bind to the operator and your camp-dependent activator protein must bind to camp site. Again, para magkaroon ng full expression of your lactose operon, dapat meron tayong repressor protein na hindi magbabind to the operator. Another requirement is that your camp dependent activator protein must bind to the cap site. Therefore, lactose must be present to prevent repressor binding and glucose must be low to allow camp to increase. Again, dapat present si lactose and dapat mababa si glucose. So, your lactose operon is an example of how transcription is set on or off. Like for example, in case of your Escherichia coli or your E. coli, which prefers glucose, but if walang glucose, it can use lactose pero dapat isynthesize muna niya si lactase enzyme. So, if a protein or enzyme is needed, magkakaroon ng transcription. So, this operon is being switched on. So, paano siya nasa-switch on if walang glucose? So, CAMP will going to increase and will going to bind to your CAP activator protein. Magkakaroon ng kink in your DNA which will going to promote transcription. Ang inhibitor na nakaharang sa operator, if wala pa ring lactose, lac operon is still off. Okay? So, if walang glucose and may lactose na, lactose removes the inhibitor sa operator. So, lac operon is now turned on. Again, full expression of your lactose operon requires your repressor protein must not bind to the operator and your CAMP-dependent activator protein must bind to your CAP site.